Good morning. Today we are going to study the algal type Chara. In our previous classes, we have discussed about the class Chlorophyceae and the different algal forms that belongs to the class Chlorophyceae. And so today in this class, we will go directly into Chara. In this screen, you can see a picture of Chara. It is a very beautiful algae, and one of the most important peculiarity of Chara is that first of all, it is a freshwater algae. That is, you can find it in your near ponds, rivers, pools, etc. And also, another name of Chara, it is known as stonewort. Why it is known as stonewort? Because the body of Chara, it is encrusted with calcium carbonate, or the whole body of Chara is covered with a covering of calcium carbonate. So hence it is also known as stonewort. This name is very important. Chara, it is otherwise known as stonewort. And another important peculiarity is that the presence of sulfur compounds in Chara, and due to the presence of sulfur compounds, the smell of Chara is like the smell of onion. That is due to the presence of sulfur compounds. And the common Indian species, you can study one or two Indian species. That is, one is Chara cylanica, and another one is Chara gracilis. Then Chara brownie. All these are Indian species commonly found in India. So remember, Chara is a freshwater algae, and it is also known as stonewort because of the presence of calcium carbonate in its body. Then we can study in detail the plant body of Chara. You have already seen the picture of Chara, and from that picture itself, it is clear that it is a macroscopic algae. Macroscopic means we can see it with our naked eye. It is macroscopic, then multicellular, profusely branched plant body. You can see different branches in its plant body, and the height of the plant body is about 20 to 30 centimeter. And another point is that the main plant body, it is divided into two. One is rhizoids for anchorage, and another is main axis. Just like stem in plants, there is a main axis in Chara, and just like root in plants, there is a rhizoid in Chara. See this is the picture, and here in this picture you can see rhizoids. The plant body is divided into two. One is rhizoids, and another is main axis. Main axis starts from here to the tip. Rhizoids and main axis. Then we can study one by one. First one about rhizoids. And the rhizoids, they are thread-like. Here you can see, isn't it? It is thread-like, and it is white in color. The color is white, just like plant root. It is thread-like, then white in color, and you can also see septa, oblique septa in the rhizoid. This is the enlarged portion of rhizoid. You can see septa here. Okay, you can notice different septa here. Then it is multicellular, uniciliate, and they are developed either from the base of the plant body or from the peripheral cells of the lower nodes. Okay, they mostly they develop from the lower side of the plant body. Then after rhizoids, the next part is main axis. And the main axis, see this picture, main axis. What is the peculiarity of main axis? Main axis is divided into different nodes and internode. This is one node, this is another node, this is another node, and in between two nodes. The first node, in between the first node and the second node, there is internode. Internode is situated between two nodes. So the main axis is mainly divided into two nodes and internodes, and in this Chara, unlike other algal forms, we are focusing by studying the nodes and internodes. What are the peculiarity of node? What are the peculiarity of internodes? And internode, first we will study internode. And internode, it consists of two types of cells. There are two types of cells in internode. First one is axial cell and the second is cortical cell. An axial cell means it is a central cell which is cylindrical in shape. It consists of an elongated central cylindrical cell. It is a central cell which is elongated and cylindrical in shape. And cortical cell means it is seen ensheathed or corticated as a layer on the outer surface of the axial cell. Axial cell is seen in the center and cortical cell outside the axial cell or as an ensheathing around the axial cell. This cortical cell, they originate from the nodal region. And what happens is that, what are the peculiarity of cortical cells? Here you can see, I have already said that they are originating from the nodal cells. Cortical cells are originating from nodal cells. So this is one node and this is another node. And cortical cell, these are the cortical cells and they originate from the nodal region. And in the cortical cells, there are two types of filaments. One is ascending filament and other is descending filaments. 50% of the cortical cell, they grow upward from the node. From here, this is one node. Some cortical cells, they grow upward from here to here. From down to up, some cortical cells grow and they are known as ascending filaments. And another 50%, they grow from up to down. They grow downward and they are known as descending filaments. So the ascending filament, they cover the lower half. See this is the lower half. So ascending filaments cover the lower half and descending filaments cover the upper half. And this is the joining point of ascending filament and descending filament. And please note that in all the species of Chara, cortical cells are not present. For example, in Chara corallina, it is ecorticate. Cortical cells are not present. But in most of the species, cortical cells are present. So the internodal region consists of mainly two types of cells. One is axial cells and the second one is cortical cells. Now we will study in detail about the nodal region. And the nodal region, it consists of two cells surrounded by 6 to 20 peripheral cells. It consists of mainly two cells and that is surrounded by 6 to 20 peripheral cells. And three types of appendages develop from each node. From each node, there are three types of appendages. That is, shoots of limited growth or primary laterals. Then secondary laterals or stipuloids, and then shoots of unlimited growth. There are three types of appendages that arise from the nodes. First one is primary lateral or the shoot of limited growth. Second one is secondary lateral or stipuloids. And third is shoot of unlimited growth. Then branches of limited growth or short laterals. They are also known as primary laterals or leaves or branchlets. And they arise from the peripheral cells of the node. They arise from the node and their growth is very limited. And it contains limited number of nodes and internodes. Then from below this is the primary lateral, and from here this is its enlarged view. Okay, enlarged view of the nodal region, and these are primary laterals. See the marking here, these are primary laterals. And from here, from below or from the basal region of the primary laterals, there arise secondary laterals, or they are also known as stipuloids. From the base of the primary lateral, secondary laterals arise and they are also known as stipuloids. Then branches of unlimited growth or long laterals, they arise from the axils of the primary laterals and they arise singly, and the growth is unlimited or indefinite growth. See here, this is shoots of unlimited growth, and where is its originating point? They arise from the axil here. All these are primary laterals, and the shoots of unlimited growth, they arise from the axils of the primary lateral. So primary lateral is otherwise known as shoot of limited growth. So shoot of unlimited growth, they arise from the axil of shoot of limited growth, and there they also consist of different nodes and internodes, and their growth is unlimited. That is, they grow indefinitely. Okay, the growth continues from here. This is also a shoot of unlimited growth, but all these, all these are shoot of limited growth or primary laterals, and those which arise from the axils of the primary laterals, they are known as shoots of unlimited growth. Hope you have understood the nodal region and internodal region. In the internodal region, we have studied two types of cells, axial cells and cortical cells. And in the nodal region, there are three types of appendages: branches of limited growth, branches of unlimited growth, and stipuloids or the secondary laterals. And the branches of limited growth, they are also known as primary laterals or the leaves. Then now we will pass on to the reproduction. Reproduction in Chara, there are two types of reproduction. First one is vegetative reproduction, and the second one is sexual reproduction. First we will study vegetative reproduction. And the vegetative reproduction, there are three types of vegetative reproduction. First one by amylum start, second one by bulbils, and third one by secondary protonema. Amylum starts, in this picture you can see amylum starts. And the amylum stars, they arise from the lower nodes of the vegetative cells. They look like stars. The shape is like star. So they are star-shaped aggregates which arise from the lower nodes of the plant bodies. You can see the originating point here. Okay, so they are the star-shaped aggregates arising from the lower nodes, and they store large amount of reserve food, and once they mature, they detach from the parent plant. They detach from here and they grow as an independent plant body. That is their function. Once they mature, they detach from here and they grow as an independent Chara. And the second one, it is bulbils. You can see here stem bulbil, and this is root bulbil. It is a small rounded tuber-like structure which develop on the rhizoids. They mostly develop on the rhizoids. In some cases, they also develop in the stem. So their appearance is small, rounded, and tuber-like. And just like amylum stars, on maturity, they also separate from the plant body and they grow as independent plant. And the third type of vegetative reproduction, it is secondary protonema. And the secondary protonema, they are tubular or filamentous structure which develop from the primary protonema or the basal cells of the rhizoid. They mainly develop from the primary protonema, and they are tubular or filamentous like structure, and they also develop from the basal cells of rhizoid. So vegetative reproduction, there are three types of vegetative reproduction. First one is amylum start, second is bulbils, and third one is secondary protonema. And now we will pass on to the sexual reproduction. And the sexual reproduction in other Chlorophyceae forms, the sexual reproduction in Chara is also an oogamous type of reproduction. And oogamous type, I hope you have remembered. We have studied in our earlier classes what is oogamous type of reproduction, what is its peculiarity. Anyway, I will explain once more what is oogamous. Oogamous means the female sex organ is larger than the male sex organ, and also the female gamete is non-motile and the male gamete is motile. That type of reproduction is known as oogamous type of reproduction. So Chara show oogamous type of reproduction. And unlike other algal forms, the sex organs of Chara, they are macroscopic. That means we can see the sex organs with our naked eye. So there are two types. One is antheridium, which is the male sex organ, and oogonium is the female sex organ. And here antheridium is known by a peculiar name, that is, it is known as globule. Just like shape, antheridium in Chara is known as globule and oogonium in Chara is known as nucleule. These two are very, very important. What is the name of male sex organ in Chara? It is globule and the female sex organ is nucleule. And another important point is that always the female sex organ is situated above the male sex organ, or the nucleule is always situated above the globule, and they are macroscopic. We can observe them with our naked eye. So remember the names. Antheridium means globule and oogonium means nucleule, and nucleule is always above globule. We can study one by one. First one, also this you can see here, this is globule and this is nucleule, and sometimes the Chara, it will be homothallic or monoecious. That means both the sex organs are seen in the same plant, and sometimes it will be heterothallic or dioecious. That means both the sex organs are seen in different plants. First we can study the globule or the antheridium. Antheridium, its shape is spherical. Just like the name indicates, the shape of antheridium is spherical. It is a large spherical. And the color, it is bright orange or red in color. Here you can see a bright orange color. A bright orange color is seen here. So it is a spherical body, red or bright orange in color. And there is a short stalk here. There is a short stalk, and that stalk is known as pedicel. And there is an outermost cell or the outermost wall, which consists of 8 large closely fitted curved cells, and they are known as shield cells. Here you can see shield cells, that is 8 large closely fitted curved cells or the shield cells, and they enclose an internal cavity. These shield cells, they protect this internal cavity. There are 8 shield cells which are closely fitted and curved, and they are known as shield cells. And the center of the shield cell, here you can observe a rod-shaped handle-like cell. In the center of the shield cell, a manubrium or a rod-shaped handle-like cell is present. And at the end of manubrium, you can see more or less rounded cells. They are known as primary capitula. And after primary capitula, you can also see some smaller rounded cells. They are known as secondary capitula. So at the end of manubrium, there is primary capitula, and after primary capitula, again rounded cells. They are known as secondary capitula. And you can also see branched or unbranched antheridial filaments. After secondary capitula, you can also see here there are numerous branched or unbranched antheridial filaments, and all these filaments contain antheridial cells or antheridium. This antheridial filaments, they are also known as spermatogenous filaments. Antheridial filaments or spermatogenous filaments are seen inside this shield cells. So, and each antheridial filament has about 250 cells, and each cell is an antheridium. And antheridium forms a biflagellate coiled and uninucleate anthrozoid. And each antheridial filament contains about 250 cells, and each cell is an antheridium, and each antheridial cell forms a biflagellate coiled and uninucleate anthrozoid. So one globule can produce about 50,000 anthrozoids. During the time of reproduction, this covering is known as shield cell, and these shield cells move apart. These antheridial filaments are exposed, and each antheridium produces anthrozoids, coiled anthrozoids, and thus they move to the female sex organ for the process of fertilization. So next we will go to the oogonium or the nucleule. And just like the globule, oogonium, it is also macroscopic, and it consists of a short stalk. And the peculiarity of oogonium is that it consists of 2 types of cells. In its body, there is 5 spirally coiled tube cells. Here, from here to here, this is a short stalk, and in the entire body, uh, there are 5 spirally twisted tube cells, except at the tip. And in the tip portion, there are 5 corona cells at the tip portion. So, tube cells, they are 5 spirally twisted, except at the tip, and in the tip portion, there is corona cell. And an egg cell or the oogonium is present in the center, which is uninucleate, and this rich in food materials like oil drops and starch grains. And also in the tip of the egg, there is a receptive spore. The receptive spore means a spore for accepting the male gamete. The tip portion, there is a receptive spore. So oogonium or the nucleus, it is macroscopic with a short stalk, and is always the nucleus is above the globule. And there are 2 type of cells, tube cells which cover the 5 spirally twisted tube cells which cover the body, and at the tip there are 5 corona cells, and egg is in the center, which is uninucleate, and also at the tip there is a receptive spore for receiving the male gamete. And at the time of fertilization, what happens is that numerous biflagellate androzoids, they liberate outside, they get liberated outside, and they swim in water for some time, and they reach nucleus. So when they reach the nucleus or the female sex organ oogonium, the tube cells, what happens is that tube cells, they elongate near the tip and forms a neck-like portion. And also in the corona cells, a narrow slit is formed, a narrow space is formed, or a slit is formed, and it facilitates the passage of androzoids. So numerous androzoids will come near the nucleus, but only one healthy androzoid or the male gamete will enter the nucleus, and it fuses with egg and forms a zygote or the oogonium. So only one androzoid will penetrate the wall and fuses with the egg and forms oogonium. So a zygote or an oogonium is formed with a single diploid nucleus. And for the germination of the zygote, this diploid nucleus, it undergoes meiosis, and as a result of meiosis, 4 haploid nuclei are formed. And later a septa is formed here, and there are two cells are formed, and the upper cell is known as lenticular cell, and the lower cell is known as basal cell. And the upper cell contains 1 haploid nuclei, and the basal cell contains 3 nuclei. And later the nuclei of the basal cell, they degenerate, and the upper nuclei of the lenticular cell, they again undergoes mitosis division. First division was meiotic division, and here it undergoes mitotic division, and a protonymal initial and a rhizoidal initial is formed. And this protonymal initial, it forms the main axis of the Chara plant body, and the rhizoidal initial, it forms rhizoids of the plant body. Thus, new plant is or new Chara thallus is formed, or a new plant body of Chara is formed by the germination of the zygote. And this is a schematic representation of the whole life cycle of Chara. So Chara is a very important type as far as your exams is concerned, and important points are that Chara is also known as stone bird. So remember that name. And also the sexual reproduction in Chara, the sex organs, globule and nucleus, and their role in sexual reproduction. All these are very, very important. And also sometimes they will ask Myelum stars, that is vegetative reproduction. Myelum stars, bulbils, secondary protonema, etc. is also important. So hope you have understood the class, and if you have any doubts, share your comments, and in the next class we will meet with another topic. Till then, bye bye, have a nice day. Thank you.